This video is going to look at split brain research and something called hemispheric lateralisation. And although that sounds a bit of a mouthful, don't worry, it's quite a simple idea to get your head around. Okay, the split brain research all links into this idea of hemispheric lateralisation. So if you look at your specification, there are two key terms you've got to know. Hemispheric lateralisation, so what it is, and how split brain research was conducted. Okay, so two main things on your spec. So the first thing we're going to look at is the AO1 for these particular ideas. And the first thing we're going to do is look at the key terms, give you some definitions for these two key terms. So hemispheric lateralisation is basically the idea that your hemisphere is your brain. Remember, we've got a right hemisphere and a left hemisphere. It's the idea that these hemispheres of the brain are not the same. They specialise in different processes. The right side of your brain controls certain things, the left side of your brain controls different things. And split brain research was research conducted on individuals who had their brains basically split in half. The part that connects the left and the right hemispheres, the corpus calcium, was split down the middle, and that resulted in people being able to explore things like language. So the first thing we're going to look at is hemispheric lateralisation. And this is the idea that each hemisphere, the left hemisphere and the right hemisphere, has functional specialisations. They both have their own functions that they're in control of. And a very basic way to look at it is the left side is all about language and speech. So you think left L, language L. The left side is for language. The right side is more to do with visual tasks, okay, and creative tasks. So you have the language side and the visual side. If we think about this idea that the left side is in charge of language and the right side is in charge of visual tasks, then how is it we can talk about things that are experienced in the right hemisphere? So things like face recognition, that's all under the control of the right hemisphere of your brain. But we need the left side in order to talk about them. So how does that actually happen? Well, the answer is that the two hemispheres are connected by something called the corpus callosum. And this corpus callosum is a bundle of nerve fibres that connects the two halves of your brain. And it allows information to be passed from the right to the left, from the left to the right, and so on. So you can do this little test if you want. You can work out whether your which side of your brain is the most dominant. Are you a left brainy or a right brainy? Okay, so you can have a little go at that. And here's a quite basic diagram showing you the functions of the left side of the brain and the right side of the brain. So the left side of the brain is in charge of things like language, science and maths. Tasks would generally be related to that side of the brain. The right side, on the other hand, is your creative or arts and music side. And right down the middle, down here, we've got our corpus callosum that links the two parts of the brain. And if you remember Barocca, he found that damage to the left side of the brain, in the Barocca area, resulted in language problems. You weren't able to speak. But damage on the right side in the same area didn't bring about those language problems. So that's the idea, that the left side is in charge of language, the right side is in charge of more creative and visual tasks. So we're going to have a quick look at split brain patients now. These were a series of experiments conducted by someone called Sperry. And what he did was he had patients who were being treated for epilepsy. And the surgeon would cut down the middle of their corpus callosum. And the idea was it would prevent those violent electrical activity that accompanies seizures. Okay, because the seizure and the electrical activity would jump from one hemisphere from the other. And that's what would kickstart the seizure. And what it basically led to was the communication between the two hemispheres being removed. So that corpus callosum was completely removed. There was no communication between the right and the left side of the brain. Remember, the right side is in charge of creative tasks, the left side is in charge of language. So they underwent the surgery and they cut this corpus callosum. So you can see the corpus callosum here in the middle. That was cut. Okay, so that main bundle of fibres connecting the left and right side of the brain were completely severed. So usually we'd see something in your left eye here. That information will go into your right side of your brain. They'd be processed by the right side of your brain. The opposite is also true here. If we see something in the right eye, it gets passed across down this blue arrow and is processed in the left side of your brain. So remember, like we said, the left hemisphere of your brain is in charge of the right side of your body. And the right hemisphere of your brain is in charge of the left side of your body. So, you can see an image here. Picture of the heart is in the right visual field. In the left visual field, they're seeing an ice cream. So, if a patient who had split brain surgery was asked, what did you see in the right visual field? Over here. They would say a heart. Okay, they can see it, and they have the language on that side of the brain in order to express what they saw. However, if they were asked about what they saw in their left visual field, they'd say nothing. Their brain's able to process this information because it's on the right side of their brain, but they haven't got the language on this side of the brain in order to say ice cream. Okay, so they're only able to express what they see if it's in the right visual field, because that's processed by the left side of the brain that controls language. Anything they see with their left eye, which is going to be processed in the right hand side of their brain, they're not able to say the word because they don't have that language centre in this side of the brain. Whereas usually we'd have our corpus callosum to link the two sides of the brain. This doesn't happen with split brain patients. So let's go through that one more time. So why does this happen? Why do they say nothing when they can clearly see the ice cream? So it's because the information presented on the left side, that left visual field, is processed in the right hemisphere of their brain. And that right hemisphere can see the picture but has no language centre, so it can't respond verbally. The left side doesn't receive the information because there's no corpus callosum, so it can't say what it's seen, whereas usually we'd be able to do that. So it's a fascinating area of research. We're going to look at a few more studies to give you a clearer picture of how these studies were actually conducted. So we're going to look at a range of different experiments on split brain research patients. And if there's anything you're still unsure of, we'll go through it in class. So remember, the information here is only presented to one hemisphere. The patient is asked to fixate on a dot in the centre of their screen. And the information is then presented to their left field or their right field of vision. And they're asked to make a response. So they can make a response with their left hand, which is obviously controlled by their right hemisphere, or their right hand, which is controlled by the left hemisphere, or they can give a verbal response, which is again controlled by their left hemisphere. Remember, L for left, L for language. So the image is presented in either field for a split second only. So there's no time for someone to look across either way. It's presented to either their right or their left visual field. And in a normal brain, the corpus callosum would share the information between the two hemispheres. But this doesn't happen with split brain patients. So it'd be a very similar setup here. Someone sits down, they fixate on this dot in the middle of the screen there. They'll be given an image or word. So you've got key on his left visual field, case on his right visual field. And again, he might be asked to verbalise his response. We might be asked to pick one of the objects here that most relates to what they're seeing. So a word is flashed up briefly in the right visual field right here, the word face. And they're asked to say what they saw. Remember, if it's presented on the right hand side, it's processed by the left hemisphere of their brain. The left hemisphere has a language centre, so they can say face, they can say what they saw. Okay, because that's the one that processes verbal and language. So next time they're presented with a word on their left visual field, same word, but because the information is processed by the right side of their brain, which doesn't have a language centre, they say they saw nothing. They're not able to verbalise that information. But they are able, you can see from the picture here, they are able to draw it. Remember, the right side is in charge of visual and creative tasks. So although they can't verbalise the information, they are able to write it out or draw it. So we can look at a range of different findings now. You should have an understanding of how Sperry's experiments were conducted. Now we can look at different types of variations of his experiment. So the first one was asking people to verbally describe what they could see. If something was shown in their right visual field, they could easily describe it. But if it was presented in their left visual field, they would report there was nothing there, that they saw nothing. So why did this occur? Well, remember that language centre is located in our left hemisphere. So any objects in the left visual field get processed by our right hemisphere. The right hemisphere doesn't have a language centre, so they couldn't verbalise their answer. So another experiment looked at recognition by touch, and it's very similar to the diagram down here, where the screen was in front of them and something would be presented to their left visual field. So remember, they can't verbalise their answer because all that information is processed by the right hemisphere. And when they were asked to select a matching object using their left hand, so remember that's still linked to their right hemisphere, they could. So you can see the man here in the bottom picture, sees the word ball, would say he saw nothing, but he could select a ball from the objects. The objects were placed behind a screen so they couldn't see them. Okay, but big screen, and he'd have to move his hand around and pick the object. And the patients could not verbally identify what they saw. Remember, there's no language centre in that right side of their brain, so they couldn't verbalise their answer. But they could understand what the object was. So that was one of the key findings for recognition by touch. They couldn't verbalise their answer, but they could recognise the object by touch. So another experiment conducted by Sperry looked at presenting different images to the different visual fields. So what they did was they presented one image to the right visual field, one image to the left visual field. And then they'd ask the person to draw what they see in their left visual field. So we've got cat in the right visual field, dog in the left visual field. They'd ask the person to draw what they'd seen in their left visual field. And the person could quite easily draw a dog. Okay, remember, the right hemisphere is in charge of creativity, so it's able to draw it quite easily. However, if they then ask them what they'd drawn, the person would say cat, because that's the image they saw on their right visual field. And their language centre in their left hemisphere would give out the word cat, because it's the word they're able to verbalise. So if you look here, another example of this experiment. They told you to look at the dot in the middle of the screen. They're then presented with different images or words to the different visual fields. HE is presented to their left visual field and ART to their right visual field. So remember, HE is going to be processed in their right hemisphere. ART is going to be processed by their left hemisphere. So what word did you see? They can say the word art because they saw that in their right visual field. Their left hemisphere is in charge of language, so they can verbalise that information. However, if they were asked, point with your hand to the words you saw, then they're able to visually pick out what they saw in their left visual field. Although they said art, they would point to HE. That's because they saw it in their left visual field. Remember, the right hemisphere is in charge of vision. And again, these images experiments continued. So in this diagram here, you've got the man who's sitting down and sees a hammer in their left visual field. But because it's in their left visual field, they can't say the word hammer, so they say, I saw nothing. Because that information is processed in the right side of their brain, it has no language centre. So using your left hand, pick up what you saw. And you can see the person would go straight for the hammer. And then, if we ask him a different image, we present an image, it's got the apple here, presented to his right visual field, and that's processed in the left side of your brain, which is in charge of language, so they say, I saw an apple. And that's what all the experiments were trying to show. That if something's presented in the right side, you could always verbally answer it. If it's presented in the left side, you can't verbalise your answer, but you could draw it, you could pick out an object linked to it. You still understand what it is, but you just can't verbalise it. So the conclusion from these experiments basically was that there is some lateralisation of function between the hemispheres. The left is in charge of language, the right seems to be in charge of visual and creative tasks. So, what can we learn from this research? Why are we learning about it? Well, we're learning that there are differences between the two hemispheres within the brain. And on top of that, the left hemisphere is in charge of speech and language. And the right hemisphere is in charge and specialises in visual spatial processing and facial recognition. And that the connectivity between the two hemispheres is as important as the functions of the left and right by themselves. So now we're going to look at the AO3 for this topic. Hopefully you understand now about split brain research. If you don't, make a note of any questions and we can go through that in our next lesson. So with this research, because it's such a big topic in biopsychology, it's good to know a few different methodological issues and a few different strengths, because that could come up as a question. Discuss 3 limitations of Sperry's split brain research. So we're going to go through a few of these now. Firstly, is the issue that the level of disconnection between the two hemispheres was different in different patients. So some had much greater degree of separation than other patients. On top of that, some of the patients had experienced drug therapy as an alternative treatment before the surgery for much longer than other patients. So they were all at different starting points. Also, the comparison groups weren't seen to be valid. They didn't contain, or the control group, contained people who had no history of epileptic seizures. So some people viewed that as not a real valid group to make comparisons to the split brain research patients. Also, the data produced was really quite artificial. If you think about it in real life, if somebody has their corpus callosum disconnected, then they're going to use both eyes to view anything in their right field and anything in their left field. They're very rarely going to use one of their visual fields at a time. And therefore, they should be able to verbalise anything that they see, because everything should be seen with both visual fields and therefore processed by both hemispheres. And finally, it was only a very, very small sample. It only contained 11 people in total. So again, reducing the validity of the research. However, there are strengths to the research as well. It was a highly scientific, specialised and standardised procedure that the patients underwent. The image projected was only projected up for 1/10 of a second, so a very, very short time. Therefore, there was no time for the patients to put their eye or move their eyes across the screen in order for both hemispheres and both visual fields to see the information. So it really meant that each hemisphere was working in isolation. And really, all of these strengths ensured that each hemisphere was only receiving the information at one time. The left visual field only saw that side, the right visual field only saw information on that side. We've got to be aware of the issues regarding generalisation of these findings as well. This is a very unusual sample of participants. We only got 11 participants who all had a history of epilepsy. And it may be the epilepsy that caused changes in the brain that influenced these findings. Finally, we're going to look at a few limitations of split brain research. It's very rarely carried out nowadays, and only a small number of people actually had this quite severe operation. And certain psychologists have argued that many of the studies conducted only have 1 or 3 participants involved in them. And the conclusions drawn often have a confounding variable or physical disorder that the participants have that make the operation necessary. So it may be down to their epilepsy rather than the corpus callosum being disconnected in the first place. And finally, not all patients have the same level of sectioning. Some have less, some have more complete sectioning, and that obviously clouds the findings and makes it far more difficult for us to get a clear answer on whether the left and right hemispheres have their own functions.